Hi, this is Focus Biology and I am Barun Prabhat. In this video, I am going to describe different proteins that are involved in muscle contractions. This is my lecture outline. Muscle proteins. Contractile proteins, myosin, actin. Regulatory proteins, tropomyosin, troponin. Structural proteins, titin, alpha-actinin, myomasin, navulin, and dystrophin. And summary of the lecture. Muscle proteins. Now, myofibrils are built from three kinds of proteins. They are contractile proteins. They are able to generate force. It means they are responsible for force generation. Second, regulatory proteins. They are responsible for on and off process of muscle contraction. Third type, structural proteins. They form a structural framework. They include navulin, titin, desmin, etc. They keep thick and thin filaments in proper alignment. Again, they are responsible for providing myofibril elasticity and extensibility. It means what extent it can stretch. Again, they are responsible for linking of myofibrils to sarcolemma and extracellular matrix. Next. Now, we will see contractile proteins. They are of two types. One is called myosin, another is actin. Myosin constitutes thick filaments, while actin constitutes thin filaments. Myosin here, this is the structure of myosin molecule. You can see it. This is a motor protein. It has a molecular weight of 520,000. It has two ends. One is called C terminus or carboxy terminus. Another is called A terminus or amino terminus. It is made up of two alpha helices. These are two alpha helices which are supercoiled and four light chains here, 1 and 2 on each alpha helices. It means it has 6 subunits, 2 heavy chains, these are alpha subunits, and 4 light chains. Here it is 1, 2, 3 and 4 light chains with 20,000 molecular weight each. Single thick filament contains about 300 myosin molecules in skeletal muscles. Next, now we will see heavy chains. This is the heavy chains. Here it is. This one. It is made up of two alpha helices. Alpha helices are wrapped around each other in a left-handed manner. right? They are coil fiber structure. This is its C terminus ends. Its diameter at C terminus end is about 2 nanometer. Alpha helix is made up of two parts. One is called tail, which is about 150 nanometer, and another is called head. It is about 17 nanometer. This region is called head. When we treat this myosin molecule with trypsin, then this myosin molecules breaks down into two fragments. One is called LMM, light meromyosin, this region, and this one is called heavy meromyosin, HMM, heavy meromyosin. If we treat this heavy meromyosin with papain, then again these fragments are formed. These are called, one is called S2 and this one is called S1, or head, myosin head. This is responsible for attaching the myosin molecule to actin filament. Here you can see clearly one light chain, another light chain, one light chain, another light chain. That is 1, 2, 3, 4. 4 light chains are associated, right? Light chains are globular domains. This globular domain have myosin subfragment 1, this one, S1 myosin head group, right? They are found at the amino terminus, right? This globular domain has two sites. One is ATP hydrolyzing site and another would be the actin binding sites, this one. Next, tails of adjacent myosins. Here this one is the tail. Tails of adjacent myosins are arranged parallel to one another and they will form shaft. Here this one is the shaft of the thick filament, this one is shaft, and you can say this one, this one. These are the heads or S1 subfragments, subunits, right? These are the S1. They protrude from the shaft in a spiral manner, and this S1 remains in contact with this thin filament. Actually 6 thin filaments surrounds one thick filament. Here it is one thick filament, and here it is one, here it is 1, 2, 3, 4, 5 and 6. So these are the 6 thin filaments that surrounds one thick filament, right? Now actin, it is the chief component of thin filament. Actually actin is made up of G actin. G stands for globular. G actin forms an actin filament called F actin. Here F stands for filamentous, filament. F stands for filament. So this is the one filament, here it is, and this is the second filament. Here it is. This F-actin twists into a helix. They form a helix. You can see here. The both, one is this, another is this. These two F-actin filament twists in a spiral manner. Here it is, right? G-actin is a globular protein. G-actin is a globular protein like this. It has two sides. One is called myosin binding site. Here I am indicating MBS for myosin binding site, and another site is the ATP hydrolyzing site. AHS. A here stands for ATP, H stands for hydrolyzing, and S stands for site. So this actin molecule has two sites. One is called MBS, myosin binding site. Another is called AHS, ATP hydrolyzing site. Again, this thin filament has polarity. One is called plus end and another is called minus end. At minus end, TPM, this is called tropomodulin protein. Tropomodulin protein is present. It stabilizes its structure. At plus end, CAPZ protein, this CAPZ protein is formed. This one, right? So this actin filament is stabilized by two proteins. One is CAPZ protein on plus end and another is tropomodulin on minus end. Actin filaments of each half sarcomere are aligned with their barbed ends linked to G lines. That is, this CAPZ end is attached to the G line. Attachment with the G disc is also supported by alpha actinin, desmin, and vimentin. These three proteins also help in associating this thin filament to the G disc. Next, now regulatory proteins. There are two. One is called tropomyosin, another is called troponin. Here you can see a rod-like structure. It actually is a thin filament. You can see this rod-like structure. This one is called tropomyosin, and this one is called troponin. Tropomyosin is a rod-like protein. It expands or covers about 7 actin subunits. It means this tropomyosin is associated with 7 different actin subunits, right? Tropomyosin fits in the groove. Here, this is the groove. As we know, the F-actin, 2F-actin is twisted around each other. So, during the twisting, a groove will form. Here it is. This is one and this is another. Here you can see this is one F-actin and this is the other F-actin. and this is the groove between them. In this groove, this tropomyosin is attached, remains associated, right? It is a rod-like structure. Now troponin, it is a globular protein that has 3 subunits. One is called troponin C, another is called troponin I, and another is called troponin T. This C subunit is called calcium binding subunit, as calcium ion binds with this subunit, and this troponin I, this subunit inhibits the binding of S1, that is myosin head, to actin molecule, and this T subunit links the whole troponin molecule to tropomyosin molecule. That is, it is responsible for linking of whole troponin molecule to tropomyosin. We can remember the function of these 3 subunits in this manner. C for calcium, you can see here is C and the first alphabet is C. Here I for I subunit. Here I for inhibition. T, this one is subunit of troponin. This T for tropomyosin. This is responsible for association of whole this troponin subunit to tropomyosin. Now the one is here you can say one is this troponin molecule, another is this. The distance between these 2 troponin molecule is about 40 nanometer. Next, now structural proteins. They are titin. It is made up of about 27,000 amino acids. To be exact, 26,926 amino acids. It is the single polypeptide, or it is the largest proteins known so far. Here this one is the titin. Here you can say this is G disc, this one zigzag structure, G disc. Here this one is titin, and this titins joins to this M line. This is what? M line. This one is M line, right? So this titin joins on one side M line and on another G discs, right, of the sarcomere. Since it is a spring like, so it can stretch and spring back without producing any harm. It is also responsible for extensibility, since it is a spring, you can say, and if it is a spring, then it can be stretched, right, and it also gives elasticity. Here this one is the another titin molecule from this G disc to M line. Now alpha actinin, this is the protein of G discs. Here you can see this is zigzag line, it is the G disc. It is made up of alpha actinin, desmin, vimentin proteins. This G disc remains associated with this. This is thin filament, associated with the thin filament, and here it is the plus end of thin filament, and this is the minus end of thin filament. Here is another protein, cap G protein, right? This alpha actinin attaches to actin molecules and to titin molecules also. Here you can say This one is titin, and here it is alpha actinin. So this alpha actinin, this is alpha actinin. This one is alpha actinin. Alpha actinin attaches to thin filament, that is actin, and it also attaches to or with the titin molecule, right? Now myomycin, it forms the M line. Here you can see this is the M line. This one is the H zone, non-overlapping zone. Here it is, non-overlapping zone of A band is called H zone, and the middle part of this H zone is traversed by M line. This M line contains paramycin, C protein and M protein. These M lines contains. Myomycin is also an important constituent of this M line. This binds to titin and links adjacent thick filaments. Here you can see this is thick filament, one thick filament, this one. This is another thick filament, this one, and this 1 and 2 thick filament is joined to M line. Again this one is one titin, this one is another titin molecule. These two titin molecules are joined in the center of S zone to M line. Next, here it is nabulin. Nabulin protein, it wraps around the entire length of thin filament, right? It means if we see here, then here this is thin filament, here it would be the nabulin protein, here it would be the nabulin protein, here it is, it will remain in this fashion, nabulin protein, right? So it wraps around the entire length of thin filament. It helps in attaching thin filaments to G discs, and this nabulin regulates the length of thin filaments during development. Actually, this nabulin or titin proteins are called molecular rulers. Nabulin controls the length of thin filament here, and for nabulin it controls the length of thin filament, Tn for thin filament, while titin controls the length of thick filament, means Tc, thick filament, right? So you can remember these two words, T for thick, Tc for thick, or T for titin, sorry, this T for titin, this Tc for thick, this Tc for thick, means thick filament, this T for titin, and for nabulin, Tn for thin. Now another protein is called dystrophin. It links, here you can see, this is the dystrophin molecule, here on both sides, dystrophin. What does dystrophin do? This dystrophin attaches this thin filament to integral membrane proteins. As you can see, this one is sarcomere, sarcolemma. This one is called sarcolemma, and this is its integral membrane proteins. This is dystrophin. This is dystrophin. It attaches thin filament to integral membrane proteins. It helps in transmitting tensions generated by sarcomeres to tendons, right? Now we will summarize our today's lecture. This is the myosin protein. Myosin protein, here it is. It is made up of two alpha helices. These helices are wrapped around one another. It is 2 nanometer in diameter. Tail from here to here, it is tail part. It is 150 nanometer. And this one is head region. It is 17 nanometer. Right? This is C terminus end and this is A terminus. At the A terminus end lies 4 light chains here. 1, 2, 3, 4. 4 light chains. Right? Now this is made up of myosin protein. Here it is actin. Actin is a polar. Actin filament. It is a polar. It is made up of G-actin. This one is called single is called G-actin. It has 2 sites. One is called ATP hydrolyzing site and another is called myosin binding site. The G-actin polymerizes and forms a filament. It is called F-actin, F for filament. And 2 F-actin wraps around each other in a helical manner. Right? Here you can see one is this one, or this one, this one, this one. Here it is another. Here it is another. So in this manner they wraps. This is the cap G protein. Here at plus end it attaches to the J disc. Here it is the TPN tropomodulin, this one. It stabilizes the minus end. Right? Now here tropomyosin and troponin. Tropomyosin is a rod-like structure. It fits into this groove. Here this one is the groove. What? This one is the groove. Here you can see this one is the groove. This groove is filled with this tropomyosin. One tropomyosin is associated with the 7 actin molecule, right? And here this is troponin. Troponin molecule has 3 subunits. One is called C subunit, one is called I, one is called T. This C subunit is calcium binding, that is it binds to calcium ion. This I subunit inhibits binding of S1 or myosin head to this actin, and this T subunit is responsible for associating the whole troponin molecule to tropomyosin. And this tropomyosin and troponin remains associated with the actin, G-actin molecule. Right? Here now this one is the titin molecule. You can see this is a spring-like structure. Is a titin molecule. This is M line. Titin molecule is actually made up of 26,926 amino acid residues. It attaches one side to M line and on another side to G disc, this zigzag structure. It is a spring-like, it is elastic, it provides extensibility to the sarcomere, right? This G disc is made up of alpha-actinin, decimine, 5-mentine. These are the proteins that helps to associate this thin filament to this G disc. Here it would be the nebulin protein that regulates the length of this thin filament. So this thick filament, this titin and this nebulin are called molecular rulers, as they regulate the length of thick filaments and thin filaments. This is M line. As you know, this region is called H zone, and H zone is traversed in the middle by M line. M line is made up of it is made up of M protein, dash M protein, and C protein, paramycin, etc. This M line. Here it is the dystrophin. Dystrophin, it is this myofibrils, and dystrophin attaches this one to this integral membrane protein. This one is called sarcolemma, that is plasma membrane of sarcomere. And dystrophin is responsible for attaching thin filament to this integral membrane proteins of the sarcolemma on either side. Thanks. If you like my video, then please subscribe it by clicking on the logo of my channel. Thank you.